Hello, everyone. My name is Matt Piercy. I'm a faculty member at Yavapai College. My academic background is in virology. I earned a PhD from the Center for Infectious Disease and Vaccinology at Arizona State. So what I'd like to do during this pandemic is create a series of short informational videos about the virus, its biology, potential treatments, and also just provide general updates about the current state of affairs with the virus and how it's affecting us all. So to start this process off, what I thought I would do was make a video that talked in very general terms about the basic biology of the COVID-19 coronavirus. So to do this, I've utilized some figures that I obtained in an article that was in the New York Times. So the link to that article is shown at the bottom of the slide there. So you can go to that source if you'd like more information. And I also want to acknowledge that the figures came from that article. I didn't make those. So to start this off, let's talk about viruses in general. So what you're looking at on this slide is a schematic of the virus, of the coronavirus. But if we back up a little bit and think about viruses in general, they're very unique. So viruses, unlike bacteria, are not alive. Technically, viruses are known as obligate intracellular parasites. And so what does that really mean? That means that a virus has to be inside one of our cells in order to replicate or to make new copies of itself. A virus outside of a cell cannot make any new copies of itself. It can't make energy. It can't reproduce. It can't move. Unless a virus is inside of a cell, it really can't do anything. That's one of the reasons that antibiotics aren't effective against viruses, because viruses are not alive. So for a virus to be able to replicate or to increase in numbers, it has to get inside one of our cells. So if you look at that picture there, there's a couple different components of the virus that are labeled. Probably the most important for this discussion is shown right here. These are the spike proteins that are found on the coronavirus. That's actually how coronaviruses got their name. If you look at those spike proteins, it looks kind of like a crown, hence coronavirus. So the purpose of those spike proteins is to enable the coronavirus to get inside of our cells where it can actually replicate. So you can think of the spike protein somewhat as being a key that's going to allow it to get inside of our cells. So if we move to the next slide, we can see how that happens. So if you look at this figure here, here's the spike proteins that we just talked about. Right? And if we look down here, this is inside one of our cells, inside the cytoplasm of one of our cells. So this right here is the plasma membrane of one of our cells. So if you look at the plasma membrane of our cells, there's going to be many different molecules that are studding the surface of that plasma membrane. One of them is this protein called ACE2. So many of our cells have ACE2 on the surface. As far as the virus is concerned, you can consider ACE2 to be a lock. So in order for the coronavirus to infect one of our cells, the spike proteins found on the coronavirus have to bind to the ACE2 proteins on the surface of our cells. That's the only way that the virus can get inside of one of our cells. And I think it's a good analogy to think of this as a key and a lock. So you can think of the virus spike protein right here as being a key that has to fit into the lock, which is the ACE2 protein on the surface of our cells. If that spike protein cannot bind to the ACE2 protein, then there's no way that the virus can get inside of one of our cells or infect one of our cells. And so remember, unless this virus gets inside of one of our cells, it can't replicate and it can't increase in number. So this concept is going to be really important in later videos when we talk about antibody treatment or how vaccines work, things like that. Because essentially, if you can prevent the binding of this spike protein to this ACE2 protein on the surface of our cells, then you're going to prevent the virus from getting inside. And ultimately, you're going to prevent the virus from being able to make new copies of itself. So a lot of treatments are geared toward preventing this interaction right here. But let's say that the virus does bind to our ACE2 proteins on the surface of our cells and gets inside. What's going to happen next? Well, what's going to happen next is inside of our cells, which is shown up here, the virus is going to release its genetic content, which is RNA in the case of coronavirus, positive sense RNA. And what it's going to do is hijack our cells' machinery. So when a virus gets inside of our cells, it basically forces our normal cellular machinery that makes proteins for us, and it forces our cells' machinery to make proteins and other components for the virus. So when a virus gets inside of our cells, it basically just takes over. And our cells stop doing their normal job, and they start doing nothing but making new copies of the virus. So one copy of the COVID-19 virus could get inside one of our cells, force that cell to make maybe millions of copies of the COVID-19 virus, and then they're going to leave the cell. And that's what's shown here. So this is inside the cytoplasm of our cells. And unfortunately, a COVID-19 virus has gotten in, made millions of copies of itself. And what you're looking at in this picture is that virus budding out or leaving the cell. So again, this is the plasma membrane of our cell here. So inside here is the cytoplasm. So this virus here is in the process of leaving our cells and going out into our extracellular fluid or extracellular matrix, the space outside of our cells. So what's going to happen if the virus infection is successful is we're going to have one virus come into a cell, but now we're going to have many, many viruses leaving. And all of these viruses, as you can see, have the spike protein. So they can go on to infect more of our cells. So you can see how the number of viruses inside of our body can increase extremely rapidly. And that has the potential to overwhelm our cells, overwhelm our immune system with pretty catastrophic results for us. But we're not defenseless against these viruses. Our body mounts an immune response whenever we're infected with a virus. So if you look up here, there's something new in the picture. So these are antibodies, right? So this is the coronavirus that we've been talking about. Here's those spike proteins. So I talked about earlier how the spike protein of the virus has to bind to the ACE2 protein in the plasma membrane of our cells. So if we could prevent that from happening, we could prevent the virus from being able to get into our cells. So what our body does, whenever something new and unidentified or non-self gets into our body makes antibodies to it. So this is an antibody. Here's an antibody here. Antibodies are proteins that we make as a function of our immune system. And what they do is bind to molecules that are foreign to our body. And of course, this virus is foreign to our body. So this antibody here will have the ability to bind to this spike protein. So if the antibody is bound to the spike protein, there's no way that that spike protein can bind to the ACE2 protein on the surface of our cells. So the antibody is going to prevent infection. You can kind of think of it, if we go back to our lock and key analogy, antibodies are almost like duct tape, right? If the spike protein is the key and it has to fit into the very specific shape of the ACE2 protein or the lock, then if we can take duct tape and wrap it around the key, there's no way that key is going into a lock. So if we can cover up the spike proteins with antibodies, then there's no way those spike proteins can bind to the ACE2 protein. So in very simplistic terms, that's how the antibodies are going to function. So these are called neutralizing antibodies because they neutralize the virus. When the antibodies bind to the spike proteins, the virus has no way of getting inside of our cells. So this is a very effective defense mechanism that our body has. But the crux of the situation is after a virus infects our body, it's going to take a couple weeks to fully start making large levels of antibody. It takes a while for that to happen. So in those 2 weeks, a lot of viruses, COVID-19 included, can cause all kinds of havoc in the body. So a lot of medical treatment is geared toward treating symptoms and kind of keeping a patient's body systems going while their body is in the process of making those antibodies. But in a best case scenario, what's going to happen after COVID-19 infects somebody, their immune system is going to detect the presence of the virus. Their body's going to make antibodies to the spike protein. Those antibodies are going to bind to the spike proteins or bind to all the coronaviruses, preventing further spread of the virus. And their immune system is going to clear their body of that virus and they're going to recover just fine. So that's the basic biology of how a coronavirus or a COVID-19 infection happens. So in some future videos, we'll talk about how we can exploit this interaction between antibodies and the spike proteins to come up with multiple types of medical treatments like antibody therapy. I'm going to make a video about vaccination, which I'll make a video about, and we'll kind of go from there. So thank you for watching.